A very good morning, everyone. Today we will see HMP Shunt. HMP Shunt is hexose monophosphate pathway and it is otherwise called as pentose phosphate pathway. And this is alternative pathway to glycolysis and also TCA cycle. Of course, oxidation of glucose takes place in this pathway also. But here the importance or significance of this pathway is no ATP are utilized or no ATP are generated during this pathway. And it is multifunctional pathways since there are several intermediates are formed in this pathway which are useful for different metabolic pathways. And if we see this pathway, it takes place in all cells of the body, but it takes place in certain tissues of the body very actively. So those tissues are adipose tissue, adrenal gland, lactating mammary gland, and also liver. So these tissues are very active in carrying out HMP shunt, and it takes place in cytosol of the cell. Because the enzymes that are required for the HMP shunt are present in cytosol. And coming to the reactions of HMP shunt, there are mainly two phases of this HMP shunt. One is oxidative phase and another one is a non-oxidative phase. We will see one by one what is going to happen in oxidative phase and another one is non-oxidative phase. First one is oxidative phase and HMP shunt starts with glucose 6-phosphate. And we know how this glucose 6-phosphate is formed. The glucose, when phosphorylated, it gives glucose 6-phosphate. That reaction takes place with the help of hexokinase or glucokinase. And this glucose 6-phosphate, the very first reaction of HMP shunt, or this is the starting material of the HMP shunt. This glucose 6-phosphate undergoes oxidation reaction and it forms 6-phosphoglucanolactone. And during this reaction, so this reaction is catalyzed by glucose 6-phosphate dehydrogenase enzyme. And this glucose 6-phosphate dehydrogenase enzyme is NADP dependent enzyme. And next reaction is hydration reaction. This 6-phosphoglucanolactone undergoes hydration and forms 6-phosphoglucanate. This 6-phosphoglucanate again undergoes oxidative decarboxylation with the help of phosphoglucanate dehydrogenase enzyme. And this is also NADP dependent enzyme. So in that way, it's going to form ribulose 5-phosphate. So this is regarding the oxidative phase of the HMP shunt. And we will see non-oxidative phase of the HMP shunt. So this ribulose 5-phosphate is there, which is the product of the oxidative phase. Here, the epimerase enzyme acts on ribulose 5-phosphate and it forms xylulose 5-phosphate. And another enzyme, ketoisomerase, acts on ribulose 5-phosphate and forms ribose 5-phosphate. So in that way, when epimerase acts on ribulose 5-phosphate, it gives xylulose 5-phosphate. When ketoisomerase acts on ribulose 5-phosphate, it forms ribose 5-phosphate. And next reaction is catalyzed by the trans-ketolase enzyme, which is dependent on TPP, that is thiamine pyrophosphate. So this trans-ketolase enzyme acts on xylulose 5-phosphate and transfers 2-carbon moiety of xylulose 5-phosphate to ribose 5-phosphate. When 2-carbon moiety from xylulose 5-phosphate is transferred to ribose 5-phosphate, this ribose 5-phosphate becomes a 7-carbon compound, that is pseudoheptulose 7-phosphate. And the remaining 3 carbons of xylulose 5-phosphate is going to be formed as glyceraldehyde 3-phosphate. So that in that reaction, we are getting pseudoheptulose 7-phosphate and then glyceraldehyde 3-phosphate. And next reaction is catalyzed by trans aldolase, which is also dependent on TPP. And here this trans-aldolase acts on pseudoheptulose 7-phosphate and transfers its 3-carbon moiety from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate. When 3 carbons are transferred from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate, it becomes a 6-carbon compound, that is fructose 6-phosphate. And the remaining 4 carbons of pseudoheptulose 7-phosphate is nothing but erythrose 4-phosphate. In that way, in this reaction, erythrose 4-phosphate and fructose 6-phosphate are formed. And what is going to happen in the next reaction? We know what is going to happen with the fructose 6-phosphate. It enters the glycolysis as well as and also TCA cycle, and further it is going to be catabolized. But how about this erythrose 4-phosphate? Again, transketolase enzyme acts on xylulose 5-phosphate and transfers its 2-carbon moiety from xylulose 5-phosphate to erythrose 4-phosphate. Then it becomes 6-carbon compound, that is nothing but fructose 6-phosphate. And the remaining thing will be like glyceraldehyde 3-phosphate. So in that way, we know glyceraldehyde 3-phosphate and also fructose 6-phosphate will enter glycolysis and TCA cycle and further oxidized. So these are the two steps where we are seeing the HMP shunt. So during this pathway, no ATPs are generated and no ATPs are utilized, but it is significant because during this pathway, some important products are formed. Those are nothing but pentoses and NADPH. And what is the importance of pentoses and what is the importance of NADPH? That we will see. In HMP shunt, these are the two main products that are formed. So pentoses importance, if we see, they are very essential for the synthesis of nucleic acids such as DNA and RNA, and also nucleotides like NAD, NAD plus and FAD, coenzyme A. So these are the nucleotides that can be synthesized with the help of these pentoses. And coming to the importance of NADPH, it is very essential for the synthesis of fatty acids, steroids and also cholesterol. And another important function is synthesis of certain amino acids which needs this enzyme glutamate dehydrogenase. And another important reaction is antioxidant reactions. They require this NADPH. Glutathione reductase is going to be formed as glutathione peroxidase, and that reaction requires, which is an antioxidant reaction, that reaction requires this NADPH. And if we see cytochrome P450 system, which is going to take place in liver, this brings about the detoxification of drugs and foreign compounds, and this happens with the help of hydroxylation reactions. These hydroxylation reactions require NADPH. And phagocytosis requires NADPH. We know what is phagocytosis. Phagocytosis is engulfment of foreign particles or microorganisms. This happens with the help of WBCs. These reactions, especially, they require NADPH. And it is also required to maintain the integrity of the RBC membrane. So that's why this is very important pathway as it generates both the pentoses and NADPH. Coming to the regulation of HMP shunt, entry of this glucose 6-phosphate will restrict or will control the concentration of the NADPH, and this NADPH is inhibitor of glucose 6-phosphate dehydrogenase, and synthesis of glucose 6-phosphate dehydrogenase enzyme is induced by the increased insulin or glucagon after a meal. So, and what is going to happen if this glucose 6-phosphate dehydrogenase deficiency happens in the body? So this deficiency is an inherited sex-linked trait, and this only means to provide, HMP shunt is the only means to provide NADPH in erythrocytes. If erythrocytes are the decreased activity of this G6PDH impairs the synthesis of NADPH, and ultimately it destroys, it damages the membrane of the RBC, and that is going to lead hemolysis, and that develops a disease that is hemolytic anemia. And another important disease that happens because of the alteration in the transketolase enzyme is Wernicke-Korsakoff syndrome. So this is also genetic disorder with which is associated with HMP shunt, an alteration in the transketolase activity that reduces its affinity with thiamine pyrophosphate. And symptoms, main symptoms of this Wernicke-Korsakoff syndrome is mental disorder, loss of memory, and partial paralysis. So this is all about HMP shunt. Just to brief summarize this HMP shunt, it is very important, though it is not going to generate ATPs, or though it is not going to utilize ATPs, but it significantly produces certain important products like the pentoses and HMP shunt. It takes place in all cells, but some of the tissues are very active in carrying out of this HMP shunt. Thank you.